Well, only 2 talks left to go, regrettably. So we're looking forward to these remaining pair of talks. And this one's by Andrew Liu from the Henry M. Gunn High School in Palo Alto, California. Andrew's going to be speaking about accounting for crosstalk between signaling pathways, identifying novel models for early and late post-transplant acute rejection. So Andrew, come on up to the stage.
Good afternoon. My name is Andrew Liu, and today I'll be presenting to you my research, Accounting for Crosstalk Between Signaling Pathways Identifies Novel Model for Early and Late Post-Transplant Acute Rejection. Organ transplantation is the first choice therapy for victims of organ failure, with more than 28,000 transplants performed annually in the US since 2005. But one major obstacle is transplant rejection. In acute rejection, or AR in particular, the recipient's immune system attacks the graft or transplant organ as a foreign pathogen, and this has inhibited graft survival for decades. Naturally, current treatments will try to suppress the immune system, but they do so indiscriminately, affecting so many immune pathways that they allow for other opportunistic infection. Thus, my research focused on this biological question: which immune pathways can be specifically targeted for treating AR and avoiding the adverse side effects? To address this question, I used a powerful computational approach called pathway analysis, where I first take samples from the disease and non-disease patients, measure the differences in each gene's expression between the two groups, obtain a list of genes that are differentially expressed between disease and non-disease, and then identify the pathways that are different, thus likely responsible for the disease. But one major problem with current pathway analysis is that no existing method can account for the interactions between pathways. In the last decade, 3 generations of methods, including overrepresentation analysis, or ORA, gene set enrichment analysis, or GSEA, and impact factor, or IF analysis, have been developed. But all of these methods treat each pathway individually before selecting which ones are significant. In a complex disease like AR, that's a huge problem because AR is exactly built on multiple pathways interacting, starting with antigen presentation at the top, required to activate multiple downstream pathways in a long and complex process before allograft destruction. Thus, I hypothesized that accounting for interpathway interactions would identify more relevant pathways than existing methods. In this example of pathways A, B, and C, current methods would only analyze within the individual boxes, ignoring that gene 1 is common between all 3 pathways. I propose to integrate these pathways into a network and recognize that when gene 4 is influencing gene 1, it's also influencing gene 2 and gene 3. To test this hypothesis, I first built a network of 171 pathways from public annotation data. Then, I collected gene expression data from renal transplant biopsies in AR and stable samples, plugged these 2 as input into 4 pathway analysis methods, the 3 existing ones I mentioned, and a novel one that I developed called interpathway impact factor, or IPIF analysis, that accounts for interpathway interactions. Then, I validated the novel pathways predicted by IPIF by first analyzing independent kidney and lung transplant data, second, performing gene deconvolution on blood microarray, and third, by correlating monocyte expression profile. This provided me evidence to propose a novel disease model for rejection. In IPIF analysis, I first calculate a perturbation factor, or PF, that quantifies each gene's significance in the disease. And that's the sum of the gene's fold change and a linear sum of PFs of upstream genes in the network. Then, I calculate an impact factor, or IF, that quantifies each pathway's significance in the disease. And that's the sum of 2 factors. First, how statistically significant is the number of differentially expressed genes on the pathway? And second, what's the sum of PFs of genes on the pathway? I used IPIF to analyze kidney transplant data along with the 3 existing methods. And here are the pathways identified significant by each method, with pathways in green known to be AR relevant according to literature or validation. First, we can compare these methods statistically. no existing method identifies any pathway as significant. In fact, the lowest false discovery rate for any pathway in any existing analysis is 0.40. In contrast, IPIF identifies 10 pathways as significant at an FDR of 0.05, demonstrating the method's statistical power. But we can also compare these methods in terms of biological relevance. No more than 3 out of the top 10 pathways picked by any existing method are actually AR relevant, whereas 100% of the IPIF predicted pathways are truly AR significant. This validates my hypothesis, and furthermore, I will show you why by first analyzing the NF-kappa B pathway, and then showing you validation of the novel P38, IL-12, and IL-23 mediated pathways predicted by IPIF. But note that this method in of itself is the first contribution of my research. NF-kappa B is very well documented in rejection, known to cause activation of T and endothelial cells, and therefore should be identified by any accurate pathway analysis technique, but the existing methods fail to do so. The reason is, as you can see in the simplified version of the pathway, that there's only one differentially expressed gene, PRKCA. But in fact, existing methods ignore that NF-kappa B is influenced by many upstream AR-relevant pathways through hundreds of individual gene-gene links that add up to substantially change the state of the pathway in rejection. This demonstrates that IPIF uniquely selects this pathway precisely because it's accounting for these inter-pathway interactions. I also validated the novel IPIF predicted pathways, first IL-12 and IL-23. These genes are known to bind to activate STAT4 and produce interferon gamma, both of which are transplant relevant and gave me reason to proceed with validation. I looked at IL-12, IL-23, and STAT4 expression in 3 independent kidney and lung datasets, and in every single case, the gene was significantly overexpressed in AR versus stable, in red at a p-value of 0.05 and green 0.10. This again shows that IPIF uniquely identified these pathways because it was accounting for the IL-12, IL-23 interaction. Moreover, there is literature validation for these pathways. P38 is essential for the production of pro-inflammatory cytokines, where IL-12 and IL-23 induce Th1 and Th17 types of immune response. These are all consistent with rejection. Interestingly, 6 of the 10 pathways predicted by IPIF are mediated by these 3 genes, which are all specifically expressed in the immune cell type monocytes. This led me to hypothesize that, in fact, monocytes and their descendants like macrophages are also significant in rejection. I validated this hypothesis in whole blood analysis in 2 ways. First, by performing gene deconvolution. I compared the expression in whole blood, neutrophils, lymphocytes, and monocytes between AR and stable samples. And in these diagrams, each node represents one sample whose closeness to another depends on how similar their expression of that category is. When we see where the AR and stable samples fall, only in monocytes are all of the AR samples clustered to one side, shown in red, and all the stable samples clustered to the other, shown in blue. In other words, monocytes are the most dramatically altered in rejection, validating my hypothesis. I also correlated these AR and stable monocyte expression profiles with normal, non-transplant monocyte microarrays from Gene Expression Omnibus. In fact, the stable and normal monocyte expressions correlate very high, around 0.7, whereas AR and normal correlate very low, around 0. Again, evidence that monocytes are dramatically altered in rejection. Moreover, because I identified this in whole blood samples taken at time of rejection, not only have I demonstrated that monocytes are significant, but they are also first actors in rejection, because this was taken at a time when bloodstream was moving to the rejection site. Therefore, I am able to propose a model where first, in early post-transplant, monocytes and their descendants like macrophages are involved in antigen presentation and the secretion of IL-12, which is known to act mainly on naive T cells and therefore induces their differentiation to attack the graft. As time goes on and memory T cells develop, I have also shown that as memory T cells develop, IL-23 can become a prime facilitator because it is known to act mainly on these types of cells. And this is consistent with the fact that as time goes on in graft rejection, the graft progressively loses its function more and more. In conclusion, my research has interdisciplinary contribution. First, a novel and widely applicable IPIF analysis that is not only more statistically and biologically powerful than any existing method in the field, but furthermore can be applied to study a wide range of diseases due to the vast availability of genomics data. And second, when I applied that to study rejection, I found a novel disease model for rejection with causal early and late rejection targets, including monocytes and the cytokines IL-12 and IL-23. In ongoing work, I am first working with researchers at Stanford to validate the disease model by testing the efficacy of IL-12 and IL-23 inhibitor in vivo. And if the test results come out positive, then this drug, which is already in phase 3 testing, could soon be used to treat rejection. Furthermore, I am working to apply IPIF to a wider range of diseases, right now lung cancer and in toxicogenomic analysis. I would like to acknowledge several people and organizations for supporting my work. First, my mentor, Dr. Purvesh Khatri at Stanford. Then, Professors Atul Butte and Mini Sarwal, who directed the labs in which I worked. The Summer Institute, the Stanford Institutes of Medical Research, for funding the National Institutes of Health, and the Siemens Foundation, College Board, and George Washington University for providing this wonderful opportunity. Thank you.